We'll discuss that. Yeah, and the malaria aging thing is kind of interesting too. I'll get to that too. So I'm going to talk a little bit about methods. So I'm going to focus a little bit about the methods we developed at Medical University of South Carolina that could be applicable to a lot of people here. Some are now established. They're for proteins. Some are for cells, and some are for single cells, and we'll get to that at the end. So all the work done here has been done by students. I actually don't do anything. So Allison did the first part. So Allison graduated a few years ago. Jake Dressman just graduated with a little help from a friend. And he did a lot of the cell imaging stuff that I'm going to talk about, along and developed the single cell imaging stuff that I'll talk about at the end. And then Furkan is a PhD student that's still in the lab, and that has developed a lot of the AI tools that we use for single cell imaging. And as Rick mentioned, we're part of a group. And so we call ourselves the Mad Lab. I'm Mehta. We have Peggy Angel, Rick Drake. So it was a question in the back at the last one of the last meetings of how do we reduce aging or reverse aging? And so I always thought, we're kind of cute together. And I always wondered if AI, everybody's taking images of AI and asking ChatGPT to join them into one picture. So I did that as a lark. I got that. So if you want to get younger, AI. I'd also tell you, this is a warning. So for me, AI, I love it. It's useful. It's great for making diagnostic algorithms and doing big data analysis. But sometimes it's not as good as you think. I wish I looked like that, by the way. So with that, you heard from Rick, who talked about the spatial imaging of tissue. And so we've done this for a long time. We ourselves in our lab have probably looked at 1,000 or 2,000 hepatocellular carcinoma tissue samples, probably another 500 cholangiocarcinoma tissue samples. And there are tons of glycans that change with the development of hepatocellular carcinoma, with cholangiocarcinoma, different glycans that change if your tumor is a p53-driven tumor, different glycans if it's a beta-catenin Wnt-driven tumor, if it's a well-differentiated, non-differentiated. There are tons of glycan changes that occur with the development of hepatocellular carcinoma. And they're fundamentally different than those that change with a cholangiocarcinoma, as opposed to an intrahepatic or a distal or a perihilar. So I think we all know in cancer, there are tons of glycan changes. They happen a ton. If you think you can find a single glycan that will be used to identify even one type of cancer, like hepatocellular carcinoma, I can tell you I don't think it works. And I've tried that, and it doesn't work very well because there's just too much heterogeneity. And that heterogeneity is driven by the genetics of the cancer. We know this, that one cancer is different than another cancer, and it's driven by the genetics of that cancer. And because of that, we know the glycans have changed. We've published that just using subtypes, but you can leave it for that. So I wanted to make a test that allowed me to use and analyze all the glycan changes on all the proteins at once. So I've done work with lectins in the past. I don't know if anybody here uses lectins. I don't like lectins, so I'll disparage lectins. I found them to be difficult. They bind things you don't think they're going to bind. There are tons of things in human serum that we don't know what they are, and they bind lectins. There are antibody responses to literally everything. Every microbial product I think you could ever think of, people with liver disease and people with many diseases will develop them. So it's an issue. And they're all lectin reactive. It's a pain. So I don't like lectins. That's just me. You may like them, but not me. So I wanted to come up with an idea to be able to look at all glycoproteins at once and look at all the glycans on all the glycoproteins at once. And it seems a little stupid, but why not? Let's try it. And it was as simple as this in my thinking. So Rick showed glycan imaging of tissue, and that's just done in an instrument. That instrument doesn't know what you're putting in it. Is it a piece of tissue? Is it a piece of tissue paper? Or is it a microarray slide? It doesn't know. It just puts coordinates on everything at a micron level. So our idea was simply this, is let's replace that piece of tissue with an antibody array. Very simple. Antibody arrays have been around since 30 years ago, 40 years ago, before the first Jenner meeting. I'd like to point that out. Been here for a long time. And so we developed this method called a glycotyper, which is very simple. You just make spot antibodies across an array, add your sample. You can decalcosylate the antibody by just spraying Pingase. We make Pingase in-house, tons of it, so very easy. You add your sample, release the glycan, image it like you would a normal imaging run, and you will get data. And you can literally do this for 10 proteins, 100 proteins, 1,000 proteins, whatever you want. It's just a matter of what you want to look at. So here's what we do. So I'm a liver guy, and so here's a sample we did where we just looked at spotted arrays. We did this by hand, and you'll see some data hopefully tomorrow where it's been improved upon by other groups. But spot by hand, and I think there are only about 16 proteins here, and you'll see signals that vary in a multi-imaging run. And then you can compare groups of cirrhotics or cancers, and you can see differences in glycan. And what you will see is you will never see something that is a phenomenal biomarker alone as a glycan. You just won't. They're all about 0.6, 0.7, 0.8, vary. But you can then use all that data because you then have a lot of data. So you can have lots of glycoproteins and lots of glycan. And with big data, you can do AI, you can use machine learning and make good algorithms. So we used it for a quick study of about 16 glycoproteins in the serum of 394 patients. And we use a machine learning algorithm in a discovery set and then in a validation set. And the sample set shown here, the discovery set came from a colleague at UCSD, University of California, San Diego, and it was about 230 odd patients. And then our external validation set came from UT Southwest and had about 165 patients. These were blinded samples. The negative with blinded samples, you're dependent upon your clinicians, and AI will cause problems when you design studies blinded sometimes, and I'll get to that in a second. But what you end up getting are a lot of glycans that change on a lot of proteins. We've looked at tissue, and most of these glycans are associated with what we see in tissue. And then you can use algorithms to identify what they are. And you can get great raw curves, 0.9 something, 0.9 something in a validation set. And honestly, that was disappointing in the validation set. We developed the algorithm in the discovery set. So these are highly complicated machine learning-based algorithms that are very specific. And when we run blinded, we realized, here's the difference. Our discovery set had primarily mid to late stage cancers, primarily driven by viral liver disease, HCV, HBV. And our validation set was primarily driven by more early stage cancers, but a lot of NASH, NAFLD, which is called MASLD. AI-based and machine learning-based algorithms are great, but they're very specific. So that's just a word of warning for myself. It's like that picture I showed you. Sometimes it can look a little bit too good to be true, but it is what it is. But we're improving this, and our plan is to try to develop this platform as a biomarker platform for hepatic cell carcinoma. And hopefully tomorrow you'll hear where other groups have taken this and improved it and made it more high throughput, put QCs, quality control things on it, and really miniaturized it. So now you can do not just 16 or 20 or 30 or 100, whatever you want. So in our group, we've been pretty good at basically imaging whatever we can image. So whether it's tissue, whether it's spotted serum, whether it's the antibody arrays I just showed you, and Peggy Angel has put down just cells. But one thing we were limited on was this idea of non-inherent immune cells. And again, when we started this, kind of what I was thinking was, people do flow cytometry, and they can characterize cells. And you could use lectins to characterize cells. And again, I don't like lectins. And so I thought there's got to be a better way to basically characterize the glycosylation of immune cells that doesn't depend upon some binding reagent like a lectin. And so we thought, quite simply, hey, I can capture an antibody, use an antibody to capture a cell. So that should theoretically work. So I should be able to capture a CD4, a CD8, a B cell, a macrophage, a monocyte, whatever, and look at its glycan and do it quite easily. And to make a very long story short, as was mentioned, and we can do two things. So I'm also somebody that favors, if you're going to look at salic acid, stabilize salic acid. So the Manfred's procedure, Rick's developed a procedure, stabilize salic acid or remove salic acid. That's just me. For some of these slides I'm going to show you, I actually remove salic acid just to simplify the profile. But my personal belief is when you're doing MALDI-based analysis, remove salic acid or stabilize salic acid. That's just me. But basically what you get are, and we used in this study to start with, Jurkat cells, a normal T cell line. And you can get good capture of Jurkat cells. We started with about 10,000 cells. You can capture those cells, get a great glycan profile. In the case, it's just a kind of like a high nanostructure. But it works very well. And you can get antibodies that are quite specific. So you can look at cells. You can get high reproducibility. Our spots have gotten a lot better. But basically, this reproducibility is now down less than 5%, but it's highly reproducible. And you do see differences between resting and activated cells. So for example, if you look at a T cell at resting and activated, you'll see the glycan profile change. You can then stimulate a cell, you can treat a cell, you can do various things, and you can monitor that change in real time and look at it as it goes on. The important thing about this procedure, and this is done with bulk cells, remember, about 10,000 cells we're capturing, is that this is not a damaging method. So you can start by looking at N-glycan on those cells in this spot, and then come back and say, okay, I want to look at lipids too, and look at lipids, and look at glycogen. So you can look at glycogen, you can look at tryptic peptides, you can look at small molecules, you can look whatever you want to look at on those captured cells. So it's true multimodal imaging analysis on those captured cells. So you can do that and get whatever data you want to get from those cells quite easily. So that's the key thing about this that's quite nice. And so I would say if anybody wants to do this on bulk, it works quite well. It's very easy. Antibodies, the CD4, CD8, B cells, things of that sort, are great because they've been used in flow cytometry for years. So all you need is an imaging mass spectrometer, and you can do this, and it's quite easy to do. But the question was always, how low can you go? That's a joke, but can you do this on a single cell? So everybody wants to know if you can do it on a single cell. And so the answer is pretty much yes, and I'll show you what the data looks like for this. And it's actually pretty simple. The workflow is getting worked out. It's submitted, but again, uses an array-based format. And we use hydrogel slides now that they can be blocked quite easily. And then you just incubate with your cell type of interest, and you can capture an array of cells. And so the hard part is this, is that, and again, we use Jurkat cells because they're a good starting cell. And ideally, you can use any cell. You can use PBMCs. I think I'll show you some data from PBMCs. We have PBMCs. You can use mixed cell populations. You can take cell homogenates. You have a piece of liver. So we've taken from livers, homogenized them, made hepatocytes, cholangiocytes, capture those cells. So if you can make homogenate in single cell suspension, you can do this. But what you'll see is like that. On the top, you see a single cell. I want that. But then below it, you see a group of cells. I don't want that. So we want to look at just the single cells. Now, what I could do, if anybody's ever used like a Brooklyn mass spectrometer, you could literally teach the instrument to circle only the single cells, and you could have a student do it, and that student will then probably come get you at the night and kill you because they've been circling thousands and thousands of little cells. Nobody wants to do that. So what we did, and this is what Furkan did, is Furkan is a bioinformatics guy, he's an AI guy. So he developed a method called SoloCell that literally, and I won't go into detail about it, but literally scans the entire slide and then identifies what cell is a single cell, what cell is a double cell, what cell is a triple cell, and so on. And then gives it coordinates that then teaches the instrument which cells to go after. So instead of having to analyze everything and waste time, it only goes after the single cells. And so what you can do there is, and it's hard to see here, but now in the little green boxes, it's identified only those single cells. And as we train the algorithm, because honestly, each slide contains 20,000 cells, if not more now, you can basically teach the instrument very well because you've generated huge amounts of data, and you can teach it very well to identify where the single cells are, and then capture them, and then teach the instrument to actually do the analysis. So hopefully this movie will play. And so what you end up getting is, oh, so they're perfect. So that's the laser firing on single cell and generating mass spectra. So literally, it goes in a random fashion, identifying just the single cells, and then generates data for that. And so this is glycan analysis. And so you're basically seeing roughly 6 cells per second. So that's our throughput right about now. You can go a little bit faster, you can go a little bit slower, but this is perfectly fine and generates good data for us. And again, this is full spectra. So you can do it with silated glycoprotein, silated in single cells, you can do it with desilated, whatever you want, but it's a full spectra. And so you can get lots of glycan on this if you want. And so just to let you know, in the time of this talk, we could have analyzed over 7,000 cells. And so far, we've analyzed about 200,000 cells already, just for practicing stuff. So it's very high throughput, very simple, and it can fit just on a little slide. Again, the key point is this. it's not destructive. It's a soft ionization. So you can do your lipids, wash off the matrix, spray Pengase, get a glycan profile, wash off the matrix, spray on glycogen, get your matrix, get your image, spray on trypsin, get a peptide image, do what you want. And then, because it's just an image cell, you have a microscope. You've actually imaged this slide. You can go back to that single cell and no bioinformatics needed. Just look at it and say, okay, what was the spectra for lipids? What was the spectra for glycan? What was the spectra for glycogen? What was the spectra for trypsin? What was the spectra for a small molecule? You can do all of that just like that. So that makes it very easy and very, I think for someone that is now scared of the AI picture, I like that personally. And then you can then start challenging this by mixing in. So we always kind of start with a stacked deck. I'm not gonna lie. So in this case, we started with the Jurket cell line, slid in a cell line called 4T1. So completely different. Jurkets are one kind of cell, murine breast cancer cell. We always compare to bulk. So the one thing I will say is when you capture a cell, the fear was we'd activate that cell or modify that cell. We don't actually see that, which is kind of weird, but we'll be doing that. But basically, you can get nice heat maps of individual cells. You can look at N-glycan. You can look at lipid. You can combine them, and you can generate nice UMaps that separate out cells very easily. So very simple single-cell glycan analysis, full glycan profiles, one slide, 20,000 cells. You can capture what you want. So, and I will tell you, there are, not unsurprisingly, good antibodies and good other binding reagents that will identify all cells. And then what you can also do, and Rick mentioned this at the end of his talk, there's a company, Ambergen, that's developed mass tags. So antibodies to various targets you'd like that you can then analyze and detect in a mass spectrometer by molding. So if you want to know, hey, is my CD4 positive T cell, is it CD445R high or low? There's an antibody for that. But then you can say, hey, is my FOX3P cell, is that got a lot of glycogen or no glycogen? Is it got this glycan or that glycan? Or does it have this tryptic peptide or that tryptic peptide? Or does it have this lipid or that lipid? You can do all of that. And so, the potential for this, I think, is quite interesting. And so it's a method, it's a development. We're now using it. Rick is going to use it probably more than anyone. But it's available for us, and I think it's quite a useful tool that I think could be of interest. Because again, the blood is not just proteins, it's cells as well. So if you want to look at everything, we can now look at all the proteins and all the cells and identify what glycans are on what proteins and what glycans are on what cells. That's what we want to do. So just a conclusion. So we use this for HCC. I'm a liver cancer guy and liver disease. But I think you can use a lot of the glycotyper, that individual protein capture and glycan analysis for other things. We've used it for fibrosis. Other people have used it for lupus, for other things, and we'll hear that. And then the method we have for bulk cell analysis, again, very simple, very easy to do, and for single cell analysis. For single cell analysis, the algorithm helps. So hopefully we'll eventually make that available to everybody soon. So it just helps in teaching the instrument. With that, a whole bunch of collaborators, and then I'm a patient hog, so we get lots of samples from patients. And I always thank the patients for giving them themselves, because nothing we do, we have thousands and thousands of patients, tissue samples, serum samples. If people didn't give of themselves for scientific research, we wouldn't have anything. So I always thank them at the end. So thank you.
In terms of actually determining where is a single cell, where is a cluster, are you doing a scan first with a scanner, or are you using the multi-laser to actually determine that?
We actually image the slide.
Okay.
We have a picture of the slide. Yep. Okay. Almost like the same thing as an imaging run. Yep.
Nice talk. I have a question. So what is the risk after you capturing your cell with the antibody and then you just do the mass spectrometry of analyzing the glycosylation of the antibody that we use for capturing?
So we generally, I decoagulate my antibody. So we make Pengase in-house. We have a lot of it. So I spray we spot the antibody. I generally do a quick ping-based reaction and get rid of that antibody. So, yeah.
That's it.
But it's, yeah, that's always concerned. Some antibodies won't, are defined, but most of them I just remove as that.
Thank you. And it's very, I think, amazing talk. So, talk about the cell to cell or single cell. Any, for example, cancer cells, let's say, from pancreatic cancer cells. So, structure of these glycans one by one is different or not?
So people have seen glycan changes in pancreatic cancer. Is it on a single cell? I assume so. If you can if you can make single cell homogenates from your organ, capturing them is easy. And so that's something we could do. If you have pancreatic cancer tissue and you want to make a single cell suspension, we can look at it. That's easy.
But so you have not got the results? No.
No, we've never done single cell pancreatic cell. I need someone to give me that sample. So yeah, so that's the issue. So we look at what we look at because we can. So I've done liver. So I've taken a liver homogenate, looked at hepatocytes, cholangiocytes, Kupfner cells, things of that sort. But if you have pancreatic cancer and want to look at that, yeah, we can do that.
Thank you.
Yeah.
Very nice talk. So can I ask one question? So with regard of these sampling, are you biased towards either looking at more at the protein surface, proteins on the surface, or in the ER, or
So
the other way around?
So we actually don't, we're not permealizing the cells. So we are primarily seeing stuff on the cell surface, but I am also convinced we are seeing stuff inside as well, because we see a lot of high mano stuff, and I always think that's inside, and man nines, which is typical.
Yeah.
So, yeah.
Thank you very much, perhaps, and thank you. And next is Joanna Gomez, who is from Porto, and talking about glycobiology of cancer.